Hey there, welcome back to Bio 231 Anatomy Physiology Lectures. We're in our last chapter of this term, and that is special senses. So we're going to go through all the special senses over the course of a couple of these lectures here, and let's go ahead and look at our lecture notes and get started. We're going to start, we go out of order as compared to the book. The book starts with vision and then goes to chemical sensations, but we kind of like to talk about the more simpler chemical sensations first, and then we'll get to vision and to hearing at the end. Taste and smell are similar in a lot of ways, and that's why this top line says that they complement each other. And you'll see in a minute some of the similarities between them. What we're looking at here is we're looking at the tongue down at the bottom of the page here, and then a taste bud within the tongue. Now there are I'm actually going to go out of order also here. Let's talk about the tongue and the taste buds really quickly. So there are three major taste bud types, the circumvallate here, the fungiform, and the foliate. Okay. Now the filiform is another papilla, but there are no taste buds in humans for this particular one. So filiform are these thread-like ones that cover the front anterior 2/3 of the tongue, and they give that textural kind of feel to your tongue if you run your teeth over it, but they don't contain any taste buds in humans. Okay, these other three though do, and so the circumvallate ones are located here as a V shape in the back, and I'm going to just kind of accentuate that V shape right now, and they zoom into one particular one right here, circumvallate papilla. And then we have the fungiform, which are the mushroom-shaped ones that are all over the tongue. And these, if you look at your tongue, that anterior portion of your tongue, you'll see the little dots, and you can see the little picture has these little dots. Those are the fungiform papillae. Okay, so these are kind of knob-like elevations on the anterior portion mostly. And then we have the foliate. And the foliate are on the sides here, posterolateral. They're these guys right there on each side. And so we can go over there as well. All right. And there's a lot of taste buds on those sides there. Okay. So now let's go back and zoom in now and look at one of these taste buds. So the taste bud is really the sensory receptor organ for taste. Remember, RC is receptor. And you can see it right here to the right. By the way, this is a page, I'll just put it right here. Both of these are on page 579. 579. Okay, so go ahead and look in your textbook there and you can see those. All right, so there are three cell types that we want to look at. We want to look at the supporting or sustentacular cells. We want to look at the gustatory or receptor cells, and then we want to look at the basal cells. Okay. Now, the basal cells are actually the stem cells of the taste buds. They first become a supportive cell, and then they develop that supportive cell into a gustatory cell. So those are, and we'll draw a little arrow here. These guys are these blue ones that you see that are on the edges on the basal side of the wall. Okay, so they're deep compared to this. This here is the opening, and this is called the little taste pore, where there's an opening, and the actual chemicals that we'll pick up will go into that taste pore and then be picked up by our receptor cells. But these basal cells next will become gustatory cells. And so gustatory ones are right here. Gustatory cells. Or no, excuse me. they will become gustatory cells, but they also are going to become supporting cells. And the supporting cells, we'll come back to gustatory in a second, are these guys right here on the ends and in between some of the cells. And what they do is they form a capsule, and the capsule then has the gustatory cells inside. So they kind of insulate the gustatory cells from each other, separate them, surround them, and support them. Now the receptor cells or gustatory cells, those have these little gustatory hairs, microvilli, and they protrude right at the edge of or through that taste pore that we saw. And on the tips of those are the chemoreceptors. And so this is the dendrite of the gustatory pathway, and we'll talk about that on the next page in a minute. All right, so those are our three different cell types: supporting, gustatory, basal. All right, so these gustatory cells will die off, and we can replace them with new ones using the basal cells. So that's good. Now, there are 5 primary tastes, and then an extra thing that we taste, kind of. So we have sweet, sour, salty, bitter. Those were the 4 primary tastes when I was a young child, and now we've added the 5th one, umami. And so I think we know probably each of these pretty well, so we'll just quickly go over them. Sweet are organic substances like sugars and amino acids that give a sweet texture and taste. Sours are acids and have a lot of hydrogen protons and in their solutions. Salties are inorganic salts and metals, so things like sodium chloride. If you were tasting a metal like copper or iron, you can actually taste that in your mouth. And then bitter things are alkaloids. Nicotine and caffeine are examples of this. And then, so those are the 4 that we had originally identified. And then we have umami. Now, umami is amino acids, but specific ones like glutamic acid and aspartic acid, or glutamate and aspartate. And these are found in a lot of very rich, meaty type things. So meats, mushrooms, cheeses, those all kind of fit into this. We'll add in meats. Mushrooms, things that have a very kind of strong, meaty, full mouthful sensation are there. Oh, I can't go down. So just know that those meats and mushrooms go along with cheese. Also, monosodium glutamate, which is used as a seasoning, as a flavor enhancer in certain cuisines, is one of these as well, because glutamate is the amino acid, and it's literally just a sodium and a glutamate attached together, and it is used to kind of enhance flavor. Some people are allergic to it, but otherwise it's not a problem. It's natural. It's a naturally occurring thing that they extract out of other foods and powderize it and add it in. And then we have an extra thing that we taste, which is water. And we have receptors that will pick up that water is in our mouth and in our throat and send that information to the hypothalamus. So that is interesting. Whoops. I swear I just clicked on that. There we go. Okay. So those are the 5 primary tastes. Now, sensitivity varying with the region of tongue. Most of this has been disproven. There's probably some generalizations here that are still true, but for the most part, we're going to not worry about different parts of the body that have those tastes on them. So we're going to just ignore that. But one thing that is important is how we activate this taste receptors, no matter if it's bitter, sour, salty, sweet, umami. The chemical substance must be dissolved in saliva. All right, so saliva is made up of mostly water, right? H2O, H2O, O. And that allows the chemicals to dissolve in the saliva and then enter the taste pores. And then the gustatory hair will pick it up. And then we will stimulate the gustatory hairs and we'll get an action potential by summation. If you remember, summation means that in this case, a single, we can either have temporal or spatial summation, and that means multiple hits either over time or to the same dendrite, same receptor, will then cause an action potential, will reach threshold. So that still is happening. Now, what are the pathways in which the gustatory these taste sensations go? And there's actually 3 different cranial nerves. Okay, so there's 3. The first being the facial nerve. And the facial nerve, you remember, cranial nerve 7, it innervates the front or anterior 2/3 of the tongue. But then we had our glossopharyngeal nerve, and remember, glosso tells us tongue, and so that's cranial nerve 9, and that innervates the back part of our tongue. And then lastly, the vagus nerve, cranial nerve 10, remember, is our big parasympathetic autonomic nerve, and it innervates the epiglottis and the pharynx, so it's going to assist with swallowing. But guess what? There are actually taste receptors back on your epiglottis and on your pharynx, so we can still taste food as it goes through that area, so that's why we see that here. All of these will send afferent impulses to the medulla, the medulla, and then it will synapse, and it will eventually get to the hypothalamus and to the primary sensory cortex, which is the gustatory area of the parietal lobe. So you remember that the parietal lobe does our sensory information. Well, this is sensory information, taste. So that's kind of the route that is taken. Now, many different things affect our perception of taste, kind of how we describe things. And if just think about these for a minute, you don't have to really memorize these. One is thermo. You might have heard me sipping some coffee a minute ago. My coffee is really hot, and so it tastes very good. As the coffee cools down and gets kind of lukewarm, some of those more bitter compounds are a little more forward, and it doesn't taste as good. Mecano, that's how something feels in your mouth. If it feels correct, like if you're having a crunchy tortilla chip, you're used to that, that's what you're expecting, that enhances your perception of the flavor. If it was soggy and you put that in your mouth, it wouldn't taste nearly as good because your expectation was crunchy and you got something kind of wet. Nociceptors are, remember, pain receptors, and so that would be things like hot sauce, hot foods would be an example. Some people really like those, some people really don't like those, so it's kind of you're either you enjoy them or don't. And then smell, and smell is really the big one that I want to talk about. Taste is about 80% smell. What we often think is taste is actually probably smell. So if you had a clogged up nose and you were eating something, you wouldn't get a full taste sensation of it. And we notice this when we're sick and we have a stuffed up nose. Things taste bland and don't taste very good. We like super salty things that have really high amounts of flavor in them during that time. Probably why chicken soup is so good when we have our cold, because it is really strong flavored and has lots of often a lot of sodium in it. And we're going to talk about smell now and kind of relate how these two are together. So olfactory epithelium and sense of smell. So now what we're looking at, so this picture here is, where is this? On page 576, right? 576. And so we'll put that in off to the side here, 576. And what are we looking at? Well, first off, remember back to when you were studying the bones, you had the ethmoid bone. And that ethmoid bone sat right at the top part of your nose. And the cribriform plate is where the olfactory cranial nerve 1 was. And that's what this is right here. This is our full olfactory bulb resting on the cribriform plate of the ethmoid bone. Okay. Now, notice there are 3 different cell types, and look at what they're called. We have the olfactory receptors, and those are down here at the bottom, all right? And they're bipolar neurons. And those guys are right here in yellow. So I'm going to actually type in all of them right now. I'm going to type in basal cell, bipolar receptor cell, and then down here put supporting cell. Okay? So the ones in yellow that you see here in the picture are the bipolar receptor cells. These are the olfactory receptors. And they respond to various odors, and notice they have cilia down here at the bottom, and that cilia is embedded in mucus. So this is mucus right here, and there are cilia where the receptors are right there. If I could spell cilia correctly. There we go. All right, now, we also said that there's supporting cells, sustentacular cells, and they are in between these different receptor cells. They make up the bulk of the epithelium. And then we have our basal cells, which are the stem cells, which are producing the olfactory receptors. So doesn't this exactly look like what we just talked about with taste? We've got a receptor cell that picks up the information. We have supporting cells that are in between and around them. And we have basal cells which replace them. And notice the basal cells, again, are these little small ones that are here at the basal part of the epithelial membrane. So the nice thing about taste and smell is you can learn one and kind of know both of them pretty well. Now and you don't need to memorize anything I'm about to say in the next moment here, but there's probably somewhere in about 15 to 30 different receptor types picking up different chemicals. But we can distinguish thousands of smells because those smells have different amounts of those things, and they trigger different ones of those at different rates. There's a thing about 7 primary smells. Those are just kind of catch-alls, and they're kind of funny names. You can kind of Google 7 primary smells if you want. Ones like putrid, I think, and they all often have a lot to do with body functions and fluids and foods. They're kind of interesting, but you don't need to memorize any of that stuff. What we want to talk about is activation of olfactory receptors. So this is interesting because the olfactory receptors, obviously, if we're getting smell, we have to have a gaseous state, meaning that the chemicals have to be volatile. They have to be in the air. So in order to be smelled first, they have to be in a gaseous state. Okay, and that's what I'm going to write here. So we say first, they have to be in a gaseous state. But then they also must be water soluble because they have to dissolve in the mucus to make contact with the olfactory cells to stimulate the receptors. Okay, so notice that with taste has to be water soluble as well. It does not have to be volatile first. Usually it's not. It's usually in a solid form, and then we break down whatever the food is, and we break little bits of the chemicals off, and they dissolve in the mucus, right? So both of these mean that they have to be liberated, and they then have to be water soluble. And this is why, like, if you were open a thing of oil or butter or some other fat soluble thing, and you tried to smell it, you wouldn't necessarily smell anything because you're not getting a lot of liberated lipids in the air, and once they get into the mucus, they don't dissolve very well in the water because they're hydrophobic. So things that are water soluble and gaseous are what we smell and taste the best. Okay? So what's the olfactory pathway? So the odor is picked up by the hair cells, and these are the cilia. Okay, first. Okay, and then we go to cranial nerve 1. So the olfactory receptor cells pass through the cribriform plate, synapse with dendrites of mitral cells in the olfactory bulb. Okay, and then the olfactory bulb goes to the olfactory tract. Remember now, tract means we're inside the CNS. And we will go 2 different places. We go 2 routes. Okay, So one route, we'll put them in a different color. One route is to the temporal lobe for direct interpretation. And this just tells us what's the sensation that we're smelling. Is it a floral smell? Is it a putrid smell? Is it whatever? And then we also go subcortically to the hypothalamus and to our limbic structures, and this is our more emotional interpretation. Is this a pleasing smell? Does this smell remind us of something from the past? Do we have a link or connection to the smell? So both of those things happen at the same time. Now, there's one other cell type that wasn't shown on that picture above that I want to mention. So we'll make a little note here that it's not shown, just so you know for later, are the granule cells. And granule cells inhibit the mitral cells. And the mitral cells, remember, were in between the olfactory receptor cell and the bulb or the tracts. And so what granule cells do is they inhibit mitral cells, which helps contribute to the adaptation process. So this is where if you keep smelling something over time, you will not fire those mitral cells anymore. Your body will adapt to it and become used to it. So you don't keep having to smell that smell over and over and over. So what will happen is then we'll only have high excitation impulses only. Sometimes the example I give in class is if you go to get your, let's say you, here in Oregon, we don't pump our own gas normally. And so the people who work at the gas station, when they go to the gas station every day, the very first gallons of gas that they pump, they're going to smell those liberated gasoline molecules really well. And then after the second or third time, their mitral cells will be inhibited by the granule cells because they keep smelling the same thing, and so their body kind of ignores it over time. However, if, let's say, they were accidentally to spill gasoline all over, the sheer amount of gasoline that's liberating into the air would be a high enough excitation impulse to re-stimulate, and they would re-smell it right away. Okay, so that's what we see there with the granule cells and how they inhibit the mitral cells to only respond to high excitation impulses. All right, so now what we're going to do is we're going to move on, and we're going to start eye and vision, and then what I think we'll do is we'll only get so far, and then we'll stop, and we'll finish eye and do hearing. So we'll kind of do a little half and half on these two lectures. So let's start with the eye, and we'll see how far we get. Okay, so first off, accessory structures. You're seeing the outside of the eye here, kind of a sagittal view, or we're looking laterally into the medial part of the eye. And this is on page 555, I believe. I'm double checking my notes here while I talk to you. Yeah, so this picture is on page 555, right? And I don't care too much about a lot of these accessory structures. I really want to go more into the eye and focus on the eye, but just a couple things we'll talk about here. First are the eyebrows. So the short, coarse hair superior to the eye, there it is right there, supplied with sebaceous glands for protection. So it's producing oils, and that lubricates the hair, and it also then allows, if sweat or any water goes down wants to go hit here, instead of it then running straight down into your eye, it will go off to the sides of your eyebrows. So that's interesting and cool. And deep to the eyebrows are right here, you can see a muscle, and that's the orbicularis oculi, and that depresses the eyebrow. Okay, and we can make a little number 1 here, and that's this one. Okay, and then we also have the corrugator muscles, which are medial, and those are right here, and those are going to help open the palpebral fissures, which we'll get to in a second. But I don't care as much about those, so let's skip those for now. And then let's look at the palpebrae. So the eyelids, right? So here's an eyelid here, here's an eyelid here, meet at the medial and lateral canthi. And the medial and lateral canthi are the corners of your eye. So if you look in the mirror and you see where your eyes kind of go and meet at the ends, these are the corners of the eye. So a medial one is in towards your nose and the lateral one is out towards your ear. And these eyelids, these palpebrae, contain also some sebaceous and pseudoodoriferous glands, and so they're producing various substances to not only help with water not going into that area, but also antibacterial things like that. And they have their own muscle called the levator palpebrae superioris, and that one is seen here, right here, not medially, but right there, above, superiorly. And so we can maybe make that one a number 2 and a number 2. Okay, so, and it does exactly what it sounds like. It elevates the superior palpebrae. And what that'll do is that'll open the palpebral fissure. Now the palpebral fissure isn't a structure, it's just a space between the upper and lower eyelid, right? So it's just a space. And if we elevate that muscle above, we will make this wider and our eyes will open more and we can close it as well. Now, there's a bunch of structures here, and I'm going to not really focus on any of them, but there's different glands and different folds and all sorts of things, and we're going to kind of skip all that. I'm not worried about talking about those for now. On the, excuse me, so this is still page 555. We just zoomed in slightly. And then right at the ends of the eyelids are the eyelashes. And they have increased innervation for protection. So if anything touches those eyelashes, usually it involves a blink response to protect your eye from particles getting into it. And then there's conjunctiva or conjunctiva. And this is the thin membrane that is on the inside of the eyelid, going from the eyelash in. And it actually is continuous with our eye ball itself. Okay, so it's a thin mucous membrane that maintains eye moisture and washes the surface of the eye as liquid is produced. And you have two different parts of it. Okay, so the palpebral conjunctiva is the part that's on the inside of the eyelid, and that makes sense. Then the bulbous or bulbar or ocular conjunctiva or conjunctiva is the part that is lining the anterior surface of the eyeball, the white part that you're seeing. Okay, so that's those two that we see there on the picture. Now, because they're continuous, they fold over on themselves like that, and then we have a little space here, and this is called conjunctival sac. And that little sac is where, like, if you were to take your finger and pull on your eyelid and look down, you'd see there's a little space there and it's a little red and you can see some blood vessels. If you got something stuck in your eye, it might be in there. If you wear contacts, maybe your contact slides into that area sometimes. Okay, so those are the recesses formed at the junction between these two areas. Now, let's talk about tears for a minute. Lacrimal apparatus. The lacrimal apparatus is what is making tears. All right, so it is a group of structures that manufacture and drain away tears. And you're producing tears all the time. We don't just produce tears when we're crying. We're producing tears to have liquid wash over the eye continually. So, where does this liquid come from? Well, most of it comes from the lacrimal glands, right? And it's located in the superior lateral portion of the orbit, and it's kind of almond shaped. And this is it right here. So, you can see one right there. Okay. There's a bunch of caniculi, little ducts. You can kind of see them there in brown right here. Those are the ducts labeled, but I won't label them. And they are releasing tears over the conjunctiva. And if you see these arrows here, you'll notice these arrows are showing the route in which the fluid moves, and eventually it will go into the lacrimal canal, and the lacrimal canal will receive this fluid through this little hole on the medial side called the puncta. And the puncta, you can actually see your own if you've never seen it before. If you take the lower lid of your eye and you just open it, I'll switch back to me here. You just take the lower lid of your eye and you open it like that, and you look in the mirror. Right there, you'll see a little dot, and you can do the same thing on the other side here and see one on the other side medially as well. But normally it's tight up against the conjunctiva, and so as fluid goes in here, the fluid will get absorbed into it, go into the lacrimal canal, and then go into what's called the nasolacrimal duct. And the nasolacrimal duct is then going to send that into the inferior nasal meatus of the nasal cavity. So what will happen is those tears will go into your nose and then mix with the mucus in your nose, and will either go down the back of your throat or will come out the front of your nose. And so this is why when we have a really good cry, for some reason, maybe you're watching a really sweet movie and there's a very touching point and you start crying and you start sobbing and you're kind of becoming overwhelmed with emotion, you're producing so many tears that some tears will drop out of your eye and run down your face, right? Our normal, what we call crying. But then a bunch of that water will go into your nose, and that's why you get so sniffly, and you become kind of a little mess as your nose is runny, and that's what's happening there. Now, you make about 1 liter a day of these tears under normal conditions, so it's quite a bit. And they're slightly basic, slightly alkaline. They have salts, they have some mucus, they have antibodies to help make sure that bacteria are not forming on our eye, and they also have lysozymes, which are bacterial enzymes that will help hopefully break down bactericidal enzymes that will break them down. It mixes with some secretions of the accessory glands, and that makes an oily solution, and that helps protect, lubricate, and moisten the eye and slow evaporation of the eye. And if you don't make enough tears, this is where we often have to go ahead and have someone put in artificial tears, right, eye drops into their eye to make sure they have enough liquid. All right, so I think we're going to go a little bit farther. We're at about 34 minutes right now, and we'll maybe do another 5 to 10 minutes. So let's now move to the eye and into the eye itself and talk about some of the structures. This picture right here is page 558. You had a similar one in your lab, and I'm not going to fill out everything here, but I do want to highlight a couple things and talk about it. And so what I'll do is I'll come back to this and fill a few things in as we talk about it on this page. So the first thing we want to look at is the different tunics. We have a fibrous tunic, a vascular tunic, and a sensory tunic. 3 different tunics. And the fibrous tunic is the outermost one, and so we're going to start there. And there's 2 parts to it, the sclera and the cornea. So let's fill those in right away. So the sclera is the white part of our eye right there. And you can see that outer fibrous tunic going around the whole thing. But then in the front, it becomes modified and it's clear, and that's our cornea. Okay, so we've got our sclera and our cornea. Now, what are the functions of this? What does it do? Well, it does a couple different things. So there's a mechanical support and protection. It forms the outer part of the eye, and so it gives the eye its shape and it protects the eye. It allows intrinsic eye muscles to attach to it, and the cornea is going to assist in focusing and allows light to go through into the inner part of our eye. So the sclera is the more posterior part, and as I said, is the white outer part of the eyeball, and it's very fibrous connective tissue, and interestingly, it's continuous with the dura mater of the brain, so it's kind of an extension of our brain. And as we said, it provides structure and protection and allows eye muscles to attach. The cornea is the more anterior portion, and this is transparent or clear, and it is avascular. Okay, so avascular means no blood vessels, no immunity, meaning there's no like white blood cells or anything there, but it is well innervated, and there are pain receptors, so if something hits our cornea, it'll make us blink, it'll make us react, we'll have a reflex response. It is transparent to light. It refracts light, meaning that the light enters and goes in at an angle, and it of course protects the inner part of the eye. And you're going to hear me say this a couple times, so I might as well do it here. This area, the cornea, is the major refractor of light in the eye, meaning that the put a little star here, and we'll put a little star here so you know that the largest angle of refraction actually occurs in the cornea, not in the lens or in any other place. And we'll talk more about that in our next lecture when we go into the physiology more. Now, at the sclera-cornea junction, there is a sinus that drains the aqueous humor. And I'm not going to label it on here, but I'll point it out. So see this little spot right here? The aqueous humor, which is right here in the front, and we'll talk about that more later, will drain eventually into those two areas right there. Okay, so that's called the scleral venous sinus, and it drains the aqueous humor. Used to be called the canal of Schlemm, which I will not refer to it as. Now, our middle layer is the vascular tunic, and there's three different parts: the choroid, the ciliary body, and then down at the bottom here, the iris. Okay, let's label those three right now. So the choroid is this dark, pigmented middle layer. So let's say choroid, right? I'm actually, while I'm here, going to label the innermost one as well, which is the retina, which is this lighter colored one, because that's all I need to put on that side there. And then we have the ciliary body and the iris. So the ciliary body is right here. Right, and below it is a thing called the suspensory ligaments. Okay, and then below the suspensory ligaments and over top of the posterior to the cornea is the iris. So we will look at that as well. And, suspensory, forgot an N, suspensory ligaments. Okay, so let's go through those next. So the choroid, that's the posterior part. It's pierced by the optic nerve. See how the optic nerve here, this is the optic nerve. See how it breaks it up. This is a thin, dark brown membrane that has melanin for the absorption of light. It's very vascular, and it supplies nutrients to the retina, which is mostly nervous tissue. In the cow's eye dissection, this didn't look dark brown, it looked black, and that's because the blood that was there has kind of turned from a red to a black. But in you, it would be more of a dark brown color. Okay, now, as that choroid comes around, it starts to thicken. So here's the choroid. They peeled it away so you can see it better. And it starts to thicken and form the ciliary body, which I also spelled wrong. I'm spelling everything wrong. Sorry, ciliary body. There we go. And the ciliary body, as I said, is very thick, and it has ciliary muscle in it. And ciliary muscle controls the shape of the lens, and this is because there are suspensory ligaments that attach the lens to the ciliary body. Okay? So they control the shape of the lens via the suspensory ligaments. Now there's another structure here, we'll do it in a different color just so we can, is the ciliary process. And this is also in the ciliary body, and what it does is it secretes aqueous humor. It makes aqueous humor, and that goes into the posterior chamber, the anterior segment of the eye in the front, and I'll talk about that a little later, okay? And then we have the iris, and the iris sits between the cornea and the lens, and it has got 2 layers of smooth muscle. And you can see here from this picture on the next page that they overlap each other, and this is on page 559 of your book, that we have a circular and we have a radial. This one right here is the circular, okay, circular, and this one on the outside here is the radial. But they are also, we can have different names for them. So the circular one, why don't we do this? Why don't we start in yellow for the circular? Circular one is the constrictor muscle, because this circular action makes the pupil smaller. Now the pupil is like the palpebral fissure. It's not an actual structure, it's just the space that allows light to enter the eye. So this dark part here is just the hole. And as you can see, when the circular muscle contracts, the hole gets small. And when the radial muscle contracts, that hole gets much bigger. And so our pupil either constricts or dilates. So the circular muscle is a constrictor, and it's on the anterior part. It constricts, and it is parasympathetically innervated. Right? Remember, parasympathetic is during our rest and relaxation mode, so we want less light to enter our eye when we're resting, so we would constrict it. The radial, which we'll do in a different color, how about blue, is the pupillary dilator, okay? And it contracts, and it actually pulls the pupil open, so it's the more posterior, it's in the back, it dilates, and it is sympathetically innervated. So when we are being, under stress, we want to dilate our eyes and see in our periphery. So if we need to escape or we need to run away or something, we need to find things. We want to see our surroundings well. And the pupil is the donut-shaped structure, the iris is the donut-shaped structure that regulates the light entering the eye, and the pupil is the hole in the very center of the iris. Okay? So hopefully that all makes sense. All right, last one we're going to do is, and then we're going to stop, is our last tunic, which is the sensory or nervous tunic, which is the retina. And you remember I wrote that one in on that picture. Let's go back and look at it real quickly. Okay, so there's the retina. The retina is in kind of this yellowish brown color. It goes all the way around the eye, except in the very front here, and is continuous with the optic nerve. Okay, so let's now talk about it for a minute. It's the innermost layer. It's in the interior and posterior position of the eye. And it's got 2 layers. The innermost layer is transparent, has millions of photoreceptors, and those photoreceptors are called rods and cones. You'll see this in just a minute. Okay. And then there's an outer, more pigmented layer. And this absorbs light. It contains phagocytes to help fight off infection. And it stores vitamin A. And vitamin A will become important in a minute. So here's the outer pigmented layer right there. You can see that written down below. And then this is the more transparent layer, clear layer here. Okay. Now, so we're looking at a cross section of the retina. So notice it says pathway of light, and there's an arrow pointing from the left to the right. So light is entering this way. Light will actually go past all these, and these are our rods and cones. Okay, I don't know if I can write rods and cones in here. Not perfect, but not bad. So, and you can kind of tell because they're shaped a little bit differently. The rods are very longer, right? And the cones that they've colored differently have this more pointy shape there, which is why they're called a cone. So you can kind of see the difference pretty easily there. So light will go past these other cells, hit the rods and cones, and then activate. Those rods and cones will change. The light will actually change something, and then we'll talk about how that happens later. And then they produce action potentials. Okay. So the rods, the longer ones there that are in purple, are much more numerous, about 20 rods for every one cone. So much more numerous. And they are specialized for dimmer light and for our peripheral vision. We have more of these on the edges of our vision, and they work much better in dimmer light, meaning smaller wavelengths less strong wavelengths of light will allow the rods to fire. And so they discriminate between shades of dark and light, but they aren't very good with things like color or very, very sharp vision, and that's where our cones come into play. Our cones are specialized for color vision and acuity or sharpness. And so we see these in the center of our vision more, and there we go. And we'll talk more about those later. Now, what happens, which is interesting, and we'll get to this in a minute, is the way that the light leaves. Actually, why don't we wait? I'll mention it now, but we'll get in our next lecture. So when light strikes these, then the light comes, the action potential, hits these, and then goes here to a bipolar cell, and then here to another cell, which becomes the ventral optic nerve. And the signal, so the light comes in one direction, and the signal goes out the other direction. Okay. And these guys here are bipolar cells, right? Remember, bipolar cells we often see in special senses. That's where we see it a lot. Now, let's talk about a couple other structures. I'm not going to highlight these on the picture, but I want to show it to you. So right here is, not there. Right here is the macula lutea, and that is this little spot that is right directly in our direct line of sight from our eye. So if I drew a straight line of going up through our pupil, it would hit right here, and it's mostly cones. All right, and it's mostly cones because that's where we will put, if we were going to, if you're reading letters on the page, you would angle your eyes so they are focused right on those letters, so you could see the sharp difference between the black of the letter and the white of the page. And so those cones are very good at picking up sharpness. If we wanted to look at color really well, we would also use this area here because they're mostly cones. So contains mostly cones. It's lateral to the optic disc or the blind spot. And the blind spot is right below, we're going back to that picture, right here where there are no receptors of any kind, there's no retina, there's blood vessels coming in and out. So we have a little tiny spot in each of our eyes where we don't see anything, but our brain fills those in because we have binocular vision, so we're getting information from the other eye about what should be there, and our brain just doesn't want to have a little black hole in our vision, so it just fills in that spot. And sometimes when your both eyes are open, if you have one eye closed, you could put a little dot on a white piece of paper and move it around, and eventually that dot will go away, and that's because it's in your blind spot, and your brain just fills in that area with the white around it, the white of the paper, so it looks like it disappears. So you can try that at home if you want. But in the middle of this macula lutea is the fovea centralis, and the fovea centralis is only cones. So it's mostly cones here, but now we're only cones. And this is the small area of the center part of the macula lutea, and it's the sharpest vision area that we have. This is right when you want to look at something very precisely. This is the area you'd focus. Okay. Now, as we said, there are bipolar cells, and those synapse with the photoreceptors, and then they go to ganglion cells, and those are these guys here. These are ganglion cells. Right there, and those will leave the optic nerve. Their axons will leave. So we have the pathway is our photoreceptors to bipolar, okay, because they are in between, and then the ganglion axons leave as the optic nerve through that optic disc where that blind spot is. Okay, now that's where we're going to stop. This was a long lecture, but we covered a bunch of material there. We got through taste, we got through smell, and we got through most of the structures of the eye. We're going to go in and do a few more structures, inner structures of the eye next, and talk about the physiology of the eye. Our last part of our second lecture will go over hearing and equilibrium, and then we'll be done. Okay? Thank you. We'll see you next time.